Okay, welcome everybody to this parallel session number 4, are in the life sciences. My name is Davide Risso. I'm going to be chairing this session, and it is my great pleasure to introduce the next 4 speakers, and I really look forward to hearing these contributed talks. Just a little bit of housekeeping first. I will so the speakers will have 15 minutes for their talks. At 1 minute mark, I will ring this bell so that they know they need to wrap up. And please address all the questions to the speaker in the chat. You'll see a message from Ricardo with you have to @ the speaker and use the hashtag Q2S and then your question. And then we will, and all the speakers will respond to the questions at the end of the session. So we'll bring them all back on stage, and we'll have a nice Q&A. So the first speaker for this panel is Moritz Hess. Moritz is a research assistant at the Institute of Medical Biometry and Statistics at the University of Freiburg. And he should be joining me on stage right now.
Hi.
Here he is. Welcome. And Moritz will talk about interpretable and accessible deep learning for omics data with R and friends. And so if you can share your screen for the presentation.
Can you see it?
Yes, I can see it. And just for everyone in the attendance, by double-clicking on the screen share, you will see it larger, and you can follow the talk. So without further ado, I think I can leave the floor to Moritz and enjoy his talk.
Okay, thank you very much. So yes, this is the title of my presentation, so thanks for coming. And so I will just go a little bit into detail what this will be about. So actually, this is our setting. So we are dealing with omics type data, in this case, single cell RNA-seq data. And these colored dots represent different cells, and we have here computed the principal components of the data. So in total, the data set has about 100 variables, so 100 dimensions, and we reduce it to 2 dimensions with principal components analysis to get a 2-dimensional representation. So this looks not too bad. So we have some groups of different cells according to color, but we would go a little bit to some more sophisticated approaches and use like maybe deep models, or in this case, deep generative models. And then we could train this model on the data and then just transform the data using the learned weight matrices from the network. And then we get the following, and we can interpret it as, okay, the network has learned some nonlinear transformation, and so can now better with few dimension capture the underlying structure. So we see the cells more nicely separated, and there are different plots of accumulated cells. So, I would now briefly explain. So we have here used a DeBolzer machine. Just briefly explain what is a DeBolzer machine. Actually, it's a generative deep model, as I mentioned before. So it's a latent variable model, and we have one layer of observed variables, called V here, and we have also multiple layers of latent variables. We can have 2 in this example. We have 2 layers, but we can of course have more. And we see a gradual reduction in the number of nodes per layer. So in the terminal layer we have only 1 node, but in this case shown above, we would at least have 2 latent nodes. So because we plotted the activation of the last 2 nodes. So in the DeBolzer machine, originally all the variables observed, the latent variables, can have 2 states or are binary distributed. So we have here the sigmoid activation function. And so we then train a DeBolzer machine unsupervised, so by maximizing the likelihood of the training data. And so if we just train the model good enough, so when we put in some training data, so we have binary training data here, And we have here some kind of, we have the correlation matrix, the covariance, and then when we can draw from this generative model, can draw samples by Gibbs sampling, and like the Markov-Chain Monte Carlo technique. And then when we look at the covariance matrix of the samples, we get, okay, we see this is quite similar to the original covariance matrix in the training data, so the network has learned the distribution. So, and I think generative models are now frequently employed for applications like single cell data and for dimensionality reduction and other purposes. But so the talk is meant to be, there was this word interpretable. So in this case, our task is now, so okay, if we now look at the data in a different form, so here we have all the expression levels of all 106 genes, and again our cells color coded here. Then we want to see now that there are different patterns observed in the genes. So we not only want a good transformation, but we want also check out which genes are actually representing the essential structure in the data. So which genes make up this clustering in these two principal dimensions. And so I would now like to present an approach to infer these genes or these patterns in genes which can discriminate different cells. So So, and how do we do it? So as I mentioned before, we have our debulker machine and we can draw samples from the network, but we can not only draw samples from the observed variables, but we also get for each sample of the observed variables also the states of the latent variables. And so by training, we trained the network so that the states of the latent variables will somehow be correlated with, of course, with the observed variables. And so we can see this example to the right. So here we have 4 hypothetical synthetic observations, and we see, okay, we have here 2 variables, the second and the fifth, and we have then the states of the terminal latent variable, U here. And we see, okay, we have a pattern, we have a correlation with the state of the terminal latent variable. So if we have 1 and 1 in the second variable, we have and 1 and 0 and 0 in the fifth, we have every time a 0 in the latent variable. And so we observe a pattern. And this pattern should be or would likely be related to different subpopulations of the data. So actually, this is the simple task we have to fulfill. We have to model the joint patterns of the activation of latent variables and observed variables. And how do we do this? So we actually use a really, I would not say old-fashioned, but it's a really model with a long tradition. We use a log linear model. What is a log linear model? It just models the observed, or the expected number of counts in a cross table. So if we have, for instance, here a 2-dimensional cross table, and we have here the states of the latent variable. In this case, we have only 1, but we could have more. And we have the state of an observed variable. Then we could sample from the network and then just fill this cross table with values and would have the counts how often these states co-occur. So, and this is of course a trivial case. So, but in the log linear model, we can model the frequency of an arbitrary number of dimensions. So we can hire a higher dimensional cross table. So of course not infinitive, but because at a certain point it would take some very long to fit the models. But we can now start with a simple 2-way cross table. And in this case, we would have the second variable already selected. So this star would correspond to the second one. And I now want to briefly explain the model. What we now evaluate is, so here we have the log of the expected number of counts in this case 3-dimensional cross table, because we are now evaluating the next variable to add to the cross table. So we have now check out this variable here, and then have a 3-dimensional cross table of this variable, and the latent variable. And what is now interesting is the last term here, which is, so these are just the marginal effects in the model, these 3 ones, and here we have interaction effects. And we are now looking, okay, given we have an interaction with the first of the second variable already in the model with the latent variable and an interaction of the second and the new variable to be selected, then is actually having this second interaction, or the third interaction term of the new variable, which is currently evaluated with the latent variable, important to explain the number of counts in our three-dimensional cross table. And then we do this with a likelihood ratio test by just comparing the reduced model with the full model, and then do this for all other variables, and then we pick up the variable with the highest likelihood ratio. And then we have a three-dimensional cross table and proceed adding another variable. And so this is the whole procedure. And so we would now like to do this with our single-cell RNA-seq data. And so since R has a rich toolbox for all kind of omics data analysis and like annotation, also and processing data, and also having access to all other statistical approaches, R is a really good platform for this. But if we look at the task view for machine learning or deep learning, actually there's only, I think, one deep learning package, or like more like a wrapper to TensorFlow, which is really, I would say, yeah, up to date. And so, but if we then do a Google Scholar search for works TensorFlow and bioconductors since 2016, we get a quite number of hits for each term. But if you look at the overlap, we only have 135 publications. That means only 135 publications actually use TensorFlow and bioconductor, and so we have bioconductor here synonymous for omics data analysis. And so if you look at the publication, there's these terms are rather mentioned independently. So in our opinion, there's quite a little gap to be filled in making deep learning analysis more suitable, or to combine R with deep learning techniques. And so the approach we use here, instead of using TensorFlow, is, and another point of TensorFlow is that it's really difficult if you would want to introduce completely new architectures. For instance, the Boltzmann machines are completely differently trained than variational autoencoders and stuff. And so therefore we would need, would be difficult to adapt. And so we went another approach. So we just got asked for some help by a friend, and this friend is in this case Julia. And you see here the performance comparison for some benchmarks, and we have here runtime where small is better. We see that Julia can have really fast performance, similar performance than C, but it's much easier to write code in Julia. So actually we wrote, or better, Stefan Lenz, who's already also presenting a talk at Friday at 10:30, wrote this Boltzmann machines package in Julia, and so would now like to use this package and also the approach to fit linear models, where we have also a fast implementation for our approach that is presented in R. And how do we do that? So we use this package, which also Stefan Lenz wrote, this Julia connector, and actually it's a really cool package because it allows to call functions from Julia directly in R. So it's not directly a wrapper, but because it's so flexible, you can just really actually call the functions from Julia within R. And so we retrieve the RNA-seq data and pre-processing in R, and then perform the modeling stuff in Julia via the Julia connector, and then in this case doing some tests if the approach worked, and did some plotting. But we could also use some other package to do pathway analysis, and then using R's-rich and bioconductor-rich environment for doing all kind of analysis stuff, this kind of analysis. So actually, so this won't be a technical talk about Julia connector. We'll find the talk at Friday at 10:30 from Stefan Lenz, who wrote this. And so coming back to our single cell example. So now we have our trained the model, the deep Boltzmann machines, on the single cell data, and then use the log linear models to extract the relevant genes. In this case, we pick 8 genes. So we define, okay, let's pick up the 8 most important genes in the data set. And then now we are interested in how well this performs. And for evaluating this, we do the following approach. So actually we have here trained the Boltzmann machine on the data, which is trained on the data. Then we draw a sample of from the network. So actually, and this sample is then annotated using the training data based on the pattern in this example. Here we have 3 selected variables, but in the real example we have 8 selected variables. But what we do is actually we're doing simple pattern matching. So we check, has this synthetic observation with 106 genes in this 8 or in this case 3 genes the same state as a given training example, and then we do a match. We match for these positions and then transfer the label of empirical observation with a perfect match to the synthetic observation. So we annotate the synthetic observation using the empirical data, and then we compare actually by plotting the whole expression data. How similar are the annotated synthetic observations, or how close are they to the empirical data from which the label has been transferred? So actually, to put it more simple, we look into we inspect the 2, in this case, 2 first principal components, and then see are samples with a given color in the same area as training data with a given color. And we see that on the next picture, and we see, okay, we have here the samples from our Volvo machine, and here the original data, and we see that actually the colors do quite well match. So actually, the synthetic observation in this area are the same area where the original data is. It means that these 8 genes are really representing the essential information about the origin of the given data. And with this, I'm already quite come to an end. So to wrap it up, so from our perspective, this approach together with Julia is a really good extension to the R framework for performing the deep learning in an interpretable way. And I will, if you would like to have more get more into details of the analysis, we have a Jupyter notebook which where you can recapitulate the whole analysis we just presented, and it's available on GitHub. And yeah, and with that, I thank you very much for your attention.
Great. Thank you so much, Moritz, for the great talk. So if you all have questions for Moritz, please add them to the chat, and then we will come back to all the speakers at the end of the session for answering any questions you might have. Thank you, Moritz. I think you can stop your screen share now.
Okay. Thanks.
Yeah. So the next speaker of this session is Federico Marini. I think you all know him a little bit, at least because he's one of the organizers for e-Rum this year. And I think I'm sure everybody will join me in thanking him and the other organizers for this great online conference. But now Federico is a speaker, not an organizer in this moment. So Federico is a postdoctoral researcher at the University Medical Center in Mainz, Germany. And in his academic life, he's been dealing with many aspects of RNA sequencing analysis. And he is a very active contributor to the Bioconductor project. Today he's going to talk about his latest project in the Bioconductor package, which is called GeneTonic. So Federico, take it away.
Okay, thank you, Davide. So thank you for the kindest words. I mean, it's so me being.
And please turn off your camera. Sorry.
Yes, sure. I just switched from this role to this other role, but anyway, stickers are up for taking them afterwards. I'll switch my camera right away so that you can see at best the presentation. Okay, you can all hear me? I guess so. So I'll give it a go. Good afternoon, everyone. I am Federico, and today I would like to talk to you about my recent new package, which is called GeneTonic. And this can help you in the analysis and the interpretation of complex transcriptomic datasets. You can find me on Twitter at FedeBioInfo, and also you can find this presentation by clicking at this on this link, or if you want, I just tweeted about it, so you can follow along or take it anytime as a souvenir for later. Let's start from the data that we have to deal with. So it's RNA sequencing, and my job just became a lot easier because both Stephanie and Moritz gave excellent introductions about it. So I cut it short, and I would say that RNA sequencing gives you a high-dimensional snapshot to quantify the transcriptomic activity of all RNA species in your sample. If you do that in many samples, so we are at the transcriptional level, so this is where the action is taking place. If we do that for a number of cells or samples, then we obtain an expression matrix, and with that we can use that to compare abundances, discover, for example, differentially expressed genes, gene signatures, which might be associated with the phenotypic differences and the experimental covariates of yours. Single cells make this a little more complex because it adds the very nice cell type, single cell resolution. And in brief, there are a wide variety of applications that exist for these data types. Sometimes they are just based on a slightly different point of view, or even exploiting the very same data, but say one of the main workflow to focus on is differential expression. And to schematize a little the typical day in the life of a bioinformatician, this could be you, for example, and this is your potential new collaboration partner that comes to you, has a very nice idea about a project, and of course you might be interested into that. And after some iterations, because the productive meeting took place and the project just was kicked off, so there might be a couple of more questions about the data, and this becomes just more than a couple, and then we can just do some analysis. And when I hear the adverb just, this is my reaction, and the just do it yourself phase is what we want to avoid, and how can we actually operate to prevent these from happening. And the answer might be also coming from so the second Marie Kondo meme of the day, and to try to spark joy again in the data interpretation of your RNA sequencing. If there's anything that I've been learning in the last years, is that we are not in this business alone. So our expertise is actually need to be combined efficiently. We have results and we have interpretation. So there's on one side there is a bioinformatician, and on the other side there is the wet lab guy that might be actually that person that has the most insider knowledge about the data that we're working on. But there's a gap to mind and to fill, or at least we want to bridge that. The thing is that we are all quite curious about seeing what's going on. So we want, of course, that our analysis has to be reproducible, because that's a very important point. But also we would like to have the beauty and commodity of exploring data interactively. So how can we actually work on these together? If you've been listening to Stephanie's talk, then I don't need to say that much about Bioconductor. And I would say that it's a place to develop software. So it's a project and a community to develop software, and also yourself as a software developer. So that's it for me, but it's also a very friendly place for biologists. So what goes on over there is that whatever toolkit that you use to generate your data, you might end up being entering Bioconductor, and you do so in many cases with this very nice class that Stephanie described as well, the so-called summarize experiment. What to do with that is that, for example, if your design is quite complex and you might get a little lost in finding out what question you want to ask with your data, you can use Explore Model Metrics for that, or you can use IC to just explore it in a very much tailored way. These two packages have been something that I've been working with Charlotte, Kevin, and Aaron, and a lot of other contributors. Otherwise, if you just go straight ahead to differential expression analysis, you might use the DaMiRseq package, which can be one of your very best friends. And if you want to use that in a seamless way, you can use the ideal package that I've been also developing. And all in all, you have a lot of tools that can help you to generate novel hypotheses and insights that you want to test on your dataset. So you could say, why bother? If we have all of these methods, where's the real bottleneck then? So we have methods and tools to do this in a very efficient way, and we have even entire workflows available that really just work out of the box and are guaranteed to work with the latest set of packages. I might be biased on this part here. So if you want to visualize your data and have better quality control, better hypothesis, and thus better answers, you should definitely check out IC and ICU to explore your dataset interactively. You can link panels and observations. And then on top of it, you have very powerful frameworks like Shiny and R Markdown that can actually be used in a very nice concert way to communicate in an interactive and reproducible way. So maybe, after all, the culprit is just data interpretation. For the fans of X-Files, the truth might just be out there, not in plain sight. So how to actually get it out? So to squeeze the juice out of your data. And in many cases, what we are trying to do is when you ask your cooperation partners, just please show me your data, then this is what you see. Tables, 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 okay, text files and tables. And some of them might be encoded in different ways, different identifiers, and it's easy actually how to imagine that it's hard to squeeze out of this some kind of interesting knowledge. So I have a recipe for you that I would like to try to test. So you might and probably should, if you have nice bioinformatic partners, your dataset that are given to you in some kind of standardized format. And no, Excel does not belong to this category. So what you're going to need is, for example, a container for your expression matrix, this DDS object, a container for the result of differential expression, a table for the result of functional arrangement analysis, this so-called pathway analysis that can give you some kind of hint of what goes on at the higher level. And we will kind of decorate this with an annotation table, which can convert, for example, identifiers which are not so really human readable to human readable gene symbols. And then you can just now choose how to have it. So if you're a James Bond fan, you can take it shaken, not stirred. Otherwise, you just have it the way you want. And there you go. So basically, this whole process described a little the process of creating this package. And yes, GeneTonic is now available on Bioconductor. You can click on that link if you check out the presentation. Otherwise, you can just Google it. And the welcome page for GeneTonic looks like this. So the first thing that you can do is that, for example, you can check out in these collapsible elements the object that you just passed as parameters. And as in many, so a shiny app is as powerful as it's usable. So I'm a strong proponent of never giving people too much unless you give them a very nice introduction what to do with your application. And the answer to this, or at least one proposal to do this, is by tours. So if you click on this button over here, and now this is just an animation because it's I challenge you to read that. But in the end, what I'm doing with these RIntro.js based tours is that I'm trying to take my user through all the components of my application and then kind of enforce this person to try out with a learning by doing approach how this application can help her or him to do what they're supposed to do. So GeneTonic gives you a lot of visual overviews. You can have, for example, this plot to just display the tabular information and also, for example, encode some other feature in a non-tabular way because visual is something really appealing. Or you can take, for example, a gene set score from all the members of each gene set and then display that by sample to plot these as a heat map. You can also take gene sets and the members of these gene sets in this kind of alternative heat map, which is encoding, again, the fold change, so the change in expression across the conditions. And, for example, display that these terms might be somehow more related because they have some members in common. You can also do the following. With this information, you take anything that looks like a similarity measure. So it can be an overlap of the gene set. It can be a semantic similarity. And then you can go through you can get a multidimensional scaling plot of your data. And you can, for example, in this case, just highlight some of these some of the most relevant genes or the ones that you want to highlight. You can also take this distance measurement and display that as a dendrogram, and then you can see that, for example, these branches that form are somehow relating together terms that have some kind of coherency. And you can see that, for example, again, we want to go from thousands of genes to hundreds of gene set to let's say, a manageable amount of, say, biological topics that you want to address, because this makes interpretation easy and or accessible. You might want, for example, to also encode the fact that a gene set might be going up or down by an aggregation of the expression change that they were having. So, for example, you might have in this data set, you have some increase in the immune response and some decrease in the DNA replication initiation, which makes sense because it was the interferon gamma treatment in macrophages. And you can try to see the decomposed contribution of each gene set with this by means of this enhanced table, so to speak. The cool thing about it is that many of these plots are actually ggplot based. So if you just ggplotly them, you can have the interactive version of them. So another efficient way to actually deal with genes and gene set is, for example, by using networks. And this graph tries to capture, for example, the interplay between these entities. And you can use your mouse to zoom in and or to click to dig deeper. So it's not just about zooming. There's a tooltip, and once you click on this, what happens is that these gene set box and this gene box basically fill up with some information that is contextual on what you have. So if you, for example, if you have a gene set, then you obtain a signature heat map over there, and if you have a gene, then you have an expression plot for this feature. If it goes a little too fast, don't worry. I'll have in brief, in a couple of minutes, just a tiny demo where you can see that a little more clearly. So you can iterate at will, of course. And you see one important feature that will be described later, which is the bookmarking in action. So another compelling way to do this is it is also a so-called enrichment map. Enrichment map are graphs of different features, and a graph that is too full can be just a hairball, but once you add interactivity back in the game, then it's at once readable and or understandable again. To bring even more light to the, okay, I'll wrap it up.
That's 3 minutes.
Okay, excellent. So I want to work on this intermediate step to actually try to, say, go from the gene level to the gene set level to the meta gene set. So I want to use this graph to, for example, to apply some community detection algorithm and find representative members of it. And I couldn't help but say pursuing the metaphor for cocktails and drinks by choosing the distill table as the name of the function. At least that's still a candidate. So if your tasks are to do this kind of thing, so check out the common themes and how they relate to gene sets and so on, and repeat, and you want to keep track of it, automating things is a very good thing. So this might not be visualized like that, but I really appreciated how this guy was trying to tackle this automated task in a very much, how to say that, analogic way. I didn't want to go this analogic way, but I took a similar approach when I wanted to introduce the bookmarking feature. The bookmarking feature is something that I can actually demonstrate live. For example, if we go to the gene and gene set graph, and then we zoom in, I can either select a gene and then click on the bookmark button, and I see that this gene has been added to the list of genes that I've been interested into. And I can also do it for the responsive to interferon gamma, and again, I can click on the bookmark. I can do it also with a left control key, so I can basically just start adding genes of interest of mine to this, and they are all marked down in this bookmarked panel. I'll just click on this. Basically, I'll hop again on the metaphor of drinks, and this will basically start the happy hour. It will generate a tailored report that focuses on the features that you've been selecting. So if once that's generated, I will show it to you. Otherwise, I'll switch back to the presentation and then say that this is the quick demo that I wanted to give you. So imagine that it's done, and now it is done. So it contains, again, the input of the things that I've been checking out. And these are the genes of interest that I wanted to focus on. I will have the expression plot for them. And then again, a nice tabbed version of the signature heat map. So I selected a couple of them. And here, for example, are the gene set that I was focused on to. I wrap it up. So the package is available on Bioconverter X on GitHub. I would like to think of this as a one-stop shop for putting together all the information and all the atomic pieces of your RNA sequencing experiments, and to make this the way to reduce the time to generate insight in your data and simplify the iteration between you and your cooperation partners. It's something that should be done in an accessible way, as well in a robust way, so interactivity and reproducibility are important players. The bookmarking and drill down is fully in the spirit of bioconductors interoperability. So if you don't see the method that actually covers the input function for your function enrichment analysis results, we can write together a new shaking function, and that's gonna be pretty much it. So that was it. This is a couple of links about the package and this presentation. And I'm sorry, I went a couple of seconds over time, so I'll just give it back to you, Davide, and without further ado, for the next speaker.
Thank you, Federico. That was a great talk. Thank you.
Thank you.
Yeah, so as before, please address your questions to Federico in the chat, and we'll now move on to the next speaker, Mattia Chiesa. And Mattia is a senior data scientist at the Centro Cardiologico Monzino. And he works in the omics unit, where he works for genomics, proteomics, and metabolomics projects. And in particular, he is also a developer of bioconductor packages. And today, he is going to talk about one of them, actually version 2 of one of them, which is called DaMiRseq. So, Mattia.
Okay, thank you, Davide. So I share my screen. Okay, and I switch off. Okay, it's okay.
We still see your.
Is it okay now? You see the first slide?
Yes, yes. Perfect.
Okay, so thank you, Davide, and I join you to thank also Federico and all the organizers for the great work behind this, the organization of this event. And today I will show you DaMiRseq, which is a bioconductor package that allow building reliable prediction models starting from high-dimensional data sets such as RNA-seq data. As Federico described before, RNA-seq, which stands for RNA sequencing, is a sequencing technology born in 12 years ago to perform gene expression study with a data-driven approach. And today, so far, RNA-seq is the most spread sequencing technology. So in this figure, you can see all the sequencing technologies developed so far. And the wider the circle, the higher the number of publications for each sequencing technologies. Today, in the era of the precision medicine, many recent publications, many recent reviews point out and stress the need of tools able to handle the huge amount of data generated by the sequencing technologies and able to produce accurate prediction rules, especially in the clinical setting, the clinical practice. However, even if the number of publications for RNA-seq or machine learning in biomedical science alone is very high, the study where these two technologies are combined are still very small. And we try to find out two possible reasons. And the first one is that today RNA-seq is mainly used for physiopathological researches, meaning that the differential statistical analysis is enough to get good results. And on the other hand, most of the tools performing machine learning does not care about the management of the omics data. So all these needs got us to develop 2 years ago the DaMiRseq package. DaMiRseq package, as you can read in the first statement, offers a TD pipeline to identify informative transcriptional biomarker from RNA-seq data in order to build classification models. It is structured in 3 main parts, so the data handling, the second is the feature selection and the classification analysis, and it could easily integrated with many R packages and can be used in several settings beside the transcriptomics, such as the metabolomics, the epigenomics, and so on. What is important is what we call the Zen of DaMiRseq. We thought DaMiRseq to drive the user in a step-by-step data evaluation. So, meaning that each step is always followed by a checkpoint able to assess the effect of the action itself. Today I will show you as Davide told us before, I will show you a major implementation of the DaMiRseq package called DaMiRseq 2.0, which extends the skills of the previous, the native version, and add the new functionalities. So briefly, now it's possible to get evaluation metrics through a cross validation, a full cross validation or a bootstrap sampling. Select the optimal model on the base of these resampling strategies, normalize and handle independent set, and perform the prediction using the best model selected. So in order to give you a representative standard usage of the package, we decided for this presentation to implement a machine learning analysis starting from the findings of our recent publication, in which, by the way, Davide, we widely use one of your bioconductor packages. And here we demonstrate that the whole blood transcriptome could retrieve information about or associated to the extent of the myocardial infarction. And we concluded this paper claiming that prediction models are needed if we want to translate our findings into the clinical applications. So the aim of this analysis is to build an accurate classification model able to distinguish MI and not MI samples by using the whole blood transcriptome obtained from RNA-seq experiments. So first of all, we collected sample exploiting the GTEC portal, which you probably know gathered the transcriptome of thousands of samples of tissues. And to do that, we select sample with a tissue equal to whole blood, with an age greater than 40 years, and obviously with the cause of death, which is myocardial infarction or not myocardial infarction. So the resulting data set was composed of 482 samples and more than 56,000 of features. First of all, we split the entire data set in a balanced learning set that we use for building the classification model, and an independent test set just for the prediction step. First of all, we normalize our data set using, in this case, the variable stabilizing transformation. And after searching for the wanted variation, we remove the effect associated to the center technical variables. So now we are ready to implement our machine learning analysis. So briefly, we will build a classification model, build a classifier, evaluate the classifier through a tenfold cross-validation, select the best model on the basis of these results, the cross-validation results, and finally, we will perform the prediction of the new data set. Regarding the feature selection and the classification step, in DaMiRseq, we have two custom implementations. The feature selection is performed in 3 consecutive steps. First of all, we remove the uninformative features by a backward variable elimination. Then we will remove the highly correlated features. And finally, we select the smallest feature set associated with a class. The classification step is performed by implementing a meta-learner classifier, an ensemble classifier, combining the prediction of several weak classifiers weighted by their reliability. Here I show you the results of this analysis. As you can see, we have the evaluation metrics for each fold, and we can, as you can see here at the end of this table, on the average, this classifier reach an accuracy equal to 92% with an average number of predictors equal to 8. So on the basis of these results, DaMiRseq allows the user to select also an optimal classification model in different ways. So here I provide you just two choices. And the first one, we will select the model with an accuracy equal to the mode of the accuracies and with the minimum number of predictors. In the second choice, we will select instead a model randomly between those with an accuracy greater than 90%. And this heat map just to show you that the two feature set could have a different discrimination power. Okay, the last step is the prediction. This is told you before, but before to implement the prediction, we developed two new functions able to normalize and adjust the data, taking into account the parameters learned during the learned in the learning set. And finally, on that, we will perform the prediction. So here I just give you the prediction for both the choices just to compare the results. So the results are pretty the same, even if the accuracy for the choice 2 is a bit greater, so 91% with compared to 90%, but with a double number of predictors. So to sum up in this presentation, I just want to give you an overview of the potential of DaMiRseq, which is a powerful tool that drives the user to implement accurate and reliable machine learning models from high throughput data, such as the omics data. DaMiRseq can be used in combination with other several tools. And what's important is that it's designed to be used in a modular way, so the user can perform specific tasks instead of the whole machine learning analysis. So I want to thank you all for your attention, and if you have any question, they are welcome. Thank you.
Thank you, Mattia. So yeah, as for the other speakers, please write the questions in the chat so that we can address them all to all the speakers at the end of the session. So, and thanks, Mattia, also for mentioning my package in your presentation.
No problem.
All right, so our next speaker is Francesca Giorgolo from Code, her work focuses on elaboration of models and statistical analysis of diverse data in different fields. In her presentation today, she will show us something on healthcare data, and in particular on wearables. And she will show us her package, Sleep Detector with the capital R, of course.
Hello, everyone. Thank you for this opportunity. And I will show you my screen. Do you see something?
Yes.
Okay. I interrupt the video. Okay. Okay. Hello, everyone. I am Francesca Giorgolo, and I work for Code, a data science company based in Pisa. And I want to show you our sleep detector package, a package that was think to be a package for extract a sleep window from raw accelerometer data. In this package, there are different kinds of functions, and the first one is a function to clean the raw accelerometer data, that in this context is a particular kind of cleaning process, because we are looking for errors in the recording process. So we want to find if there are some pauses in recording, and we call them gaps, or if there are some errors, we name it overlaps of the signals, because maybe there are multiple values for the accelerometer data for the same timestamp. The main function of this package is a function to detect the inactivity and the sleep windows. And it has also a parameter to identify the non-wearing periods, so the user can choose how to handle them during the elaboration. And finally, there's also a function to visualize the results obtained with the main function. Now I will show you an example of usage of this package, and I prepare it with a dataset coming from a 3-axis accelerometer data. And here we have the columns of the accelerometer that are in milli-G units, and the timestamp column that in my case contains 1 record every 4 seconds. But the package can handle also time series with a sampling frequency higher than 1 Hertz. The first step is to use the function identify gap overlap. That is a function that needs the raw data and the column important for the elaboration. And here I can also specify the theoretical sampling frequency of my data. In this case is 0.25 Hertz. This function returns a list of 2 tables. The first one contains the original dataset with other columns, such as gap group, overlap group, and another column called drop device. These columns are useful to the user to split the whole time series into distinct blocks of acquisition, and also to filter away if some errors in the recording happened using the drop device column. The second table instead is a summary table, and it looks like this table, where you can find those columns. And for example, here I can see that there's a huge gap in my recording process, because in fact we can see that the last timestamp is on the 16th of August, and the first one here is on the 4th of October. So maybe it can be useful to split the whole time series into 2 acquisition blocks using the gap group column. Looking into the first acquisition block, we can see also different overlap groups. And in particular, we can see that in this window, we have the theoretical sampling period, but also we have this 0. That means that in this period, the accelerometer data is doubled. So we have to clean it. When the parameter plot is set to true, the function returns also a plot where we can focus on the gap group 0 overlap group 1, where we found the anomalies in the sampling periods, And here we can see the 3 traces of the accelerometer. And those in red are the signals labeled as to drop. This label is made up using the drop device column I mentioned before. And the not separated label here is not present, but is useful when the function is not capable to handle the different kind of signals that found in that time window, and so the user maybe have to check personally what happened and decide what to do with this time window. And it can be useful, maybe look at this table to identify the range instead of scrolling all the time series. After that, I split the whole acquisition data using the gap group column, and in each acquisition block, I only retain the records labeled as 0, so the to keep signals. In the first acquisition block, I will try to extract the sleep window using the detect SPT function that is the main function of the package. Here you can find some parameters. The 2G conversion factor is a parameter that lets the user to suggest in which unit is recorded the signal. In my case, the accelerometer data are in milli-G, so I have to provide as factor 10 raised to -3. And below there are 2 other parameters, threshold and use nowhere, that enable the user to choose how the threshold between activity and inactivity should be calculated. The detect SPT function returns a list of 4 tables. The first 3 tables are intermediate results tables that are useful to understand what was happened during the elaboration process, and maybe they are also useful to make different kinds of plot. Instead, the last table is a table with the inactivity and the sleep window for each day of acquisition. In particular, we have the starting time, the ending time, and the duration of the inactivity window. Note that the time here is midday, because we are looking for sleep windows, so we focus on the night hours, and the usual day, the midnight to midnight day, is not so appropriate, because it would split the sleep window into 2 parts. So we use this convention that a day is a noon to noon day. Finally, the plot SPT function provides a tool to visualize what was detected by the detect SPT function. The default visualization is the rolling median visualization, that is an elaboration of the raw signal of the accelerometer. And here we can see the sleep window and the inactivity windows found in this night, Thursday night. Here you can see the activity windows in red and the rest windows in light or dark green, depending on their duration. The inactivity and sleep windows are calculated starting from those windows and unite them when the activity periods between them is is short. In fact, this let us to handle the discontinuities in the sleep. For example, when someone goes to the bed, to the bathroom, or goes to drink something during the night, and then comes back to the bed. And another visualization we can also retrieve is this kind of visualization. So we can provide the function, the raw data set, and also a trend column. In my case, I choose the y-axis of the accelerometer. And so I can see the trend of the raw data on the detected windows of inactivity and sleep. Thank you for your attention, and follow us on our socials. And if you have some questions, you can ask me later. Thank you very much.
Francesca, and for the very nice presentation. And thank you all of the speakers for this great session. I think if you can stop your screen share, Francesca, and everybody else can join me on stage, we can proceed with the Q&A.
Okay.
Great. So I guess we will just go, top to bottom from the first talk. So we have about maybe 20 minutes for Q&A. So I think we have time to answer a few questions. So maybe I'm gonna do a round, a full round of one question each, and then we will circulate so that we're sure that everybody has, a few questions to answer to. So we'll start with Moritz. And this question comes from Patricia Reiser-Welch. And she says, interesting use of TensorFlow. Would other machine learning algorithms achieve the same results? Have you considered using a form of genetic algorithms or genetic programming?
Okay, thanks for the question. So maybe I made it not clear in the talk. So we were not using TensorFlow, but we were using our own implementation of Boltzmann machines written in Julia and via the Julia connector. So and for the other question, we did not. Yeah, we used some basic like linear approaches like linear regression. Like for instance, like boosting to or boosted linear models, like to infer essential variables, but actually no, not with other approaches like variational autoencoders, and yeah, not with other approaches.
Yeah, thank you. So not to just immediately do differently from what I said, but since there's a similar question, maybe we can do a couple per speaker. And actually, there are two questions that are kind of similar. One is from Gianmarco Franceschini, who says, can you comment on the choice of Boltzmann machine as generative model compared to VAE or regularized versions of the deterministic AE? And Britta Velten also says, where do you see advantages or shortcomings of Boltzmann machines versus variational autoencoders? So basically, how do you compare with autoencoders?
Okay, so we choose Boltzmann machines because from our experiment they worked out quite well. So in this case we by choice we choose a rather small data set with only 1,500 observations because would like to address this area, because frequently experiments are still conducted with few amount of cells in this example, and in this small sample setting from our experiments, the Boltzmann machines really turned out quite better than variational autoencoders, specifically so with autoencoders rather from our experiments, tended to too strongly regularize, so that we actually they failed in capturing the different subpopulations, but rather rather not getting this multimodal structure so well. So it's maybe also related to the prior distribution used in the autoencoders, but this is actually why we use deep Boltzmann machines in this example.
And I guess the second part of the question from Britta Velten was, what about linear latent variable models?
Mm-hmm. So for sure, so what we did here, we also relatively strongly selected, pre-selected a subset of genes. So actually, because otherwise would get no meaningful results, and so that's actually the disadvantages and advantages. So actually, we can, compared to linear models, much better if there are interactions between variables, better like blocks of genes which only occur together, much better model these interactions. But for sure, we need more better relationship between variables and training examples. So, and I think that there's a trade-off if you have not too many training examples, but relatively few variables. And of course, I think the linear approaches would work better. But to get these networks should get the directions between variables with non-linear dependencies better.
All right. Yeah, thank you. So I guess do a couple of questions for Federico. So Michal Schwiktek, I'm sorry, I'm butchering your name, I'm sure, asks, is it possible to set the digital environment in GeneTonic or simply bioconductor for transcriptional factors of genes to find out how their function different?
Okay. I hope that I don't misunderstand the question. So it might be something that you probably might be focused on single gene set signatures. So I think one thing that I was thinking of is, for example, to adding custom signatures, for example, also to compare, for example, in the gene set graph, how these might relate and how these might be somehow interacting with the existing ones that are returned by the enrichment results. Otherwise, you might be always subsetting your, say, expression matrix in advance and then give a more thorough look to that. I hope that clears up the question, but in principle, yes, you can. If not now, it's some feature that I already was thinking to actually implement in the next feature.
Yeah, so +1 on that feature request, because I think looking at transcription factors is very interesting.
Yeah.
Most of my collaborators ask specifically for that.
As a golden rule, I have the critical mass of 3 for making a feature request a feature. So if there's another +1.
Yeah, just put it in the chat if there's another +1. The second question for you, Federico, Enrico Ferrero asks, does GeneTonic support single cell data, or is it specifically designed with bulk in mind?
So designed it was with bulk, because that's actually the case that we wanted to cover most, because that's the one that actually most of the time our collaboration partners want more and more iterations with, even if it's just bulk. But in principle, I wouldn't exclude that that it could scale up to single cell. I've never tried actually to have a DDS object actually contain a very large single cell experiment data set, but in principle you could. I'm seeing that, for example, might be some more efficient way to deal with that, but it's something that I can definitely work on. If you want to +1 on that as well.
Okay, so I guess a couple of questions for Mattia. The first one is from Jeremy Selva, and they ask if DaMiRseq can be used in lipidomics data as well. I have to say I don't know lipidomics data, but
Yeah, the answer is absolutely yes. As I told you before, DaMiRseq is structured in 3 parts. So the first one, the normalization, is dedicated only for RNA-seq, but the second and the third parts, the feature selection and classification analysis, is for any kind of omics. So you just to import a normalized data set, and then you can use the functions for the feature selection and the classification analysis. So absolutely yes.
Okay, thank you very much. And the second question for you is from Bruce Burnett. Have you looked at time series where rate of change in a blood test value is more useful than a single value?
So the short answer is no, I have not, because the data I present you just a toy data set, so it is just an example data set. So I can try. I think no problem with time series data, because as for the previous question, every time you can import your data inside the DaMiRseq, and your aim is to develop a machine learning analysis, no problem with time series data. So I didn't try, but for lipidomics I tried, so it worked. But for time series data, I didn't try yet. So I think yes, but I didn't try yet.
Okay, thank you.
You're welcome.
So next question is from Francesca, and it's from Eric Wanzhou. I hope I'm pronouncing it right. Was the sensor data being sent remotely and then analyzed later? If so, what technology, e.g. LoRaWAN, did you use?
Okay, the data I have was sent for a post elaboration, so it's not an online process, but I didn't deal personally with the transmission of the data, only on the algorithm part, so I don't know how was the technology behind it.
Okay, and there's a follow-up question by the same person. Could machine learning be used to identify the active or sleep times?
Yes, but there can be some problems, because when a person is not very active, or maybe if his sleep is frequently interrupted from small activities, or is not so good sleep, maybe points, records representing this timestamp will be mixed one with each other, so the sleep windows is not so clear.
Mm-hmm.
I have a follow-up question on this, Francesca. If
I think we should circle back to Moritz.
Yeah.
And I think this is a question I also had. So Oystein Sorensen asks if Keras, which can be used with different backends, could be an alternative for running deep learning in R, and thinking particularly about the R package called Keras.
Mm-hmm. Well, actually, we had not in our group exactly, but at the institute we had some project with literature mining, and this was run in R via Keras, and Keras is also an interface to TensorFlow. So we have like 3 layers, and from my experience, no, not from my experience, but from what I heard, it was really complicated to get all this stuff running, because we have to adjust everything together. So okay, here we also have to have a Julia version running, but we have 1 layer less. So actually, I think the thing what applies to TensorFlow would also apply, I guess, to this Keras thing. Even it's even more. We have again 1 layer added to the whole stack to finally get through to the algorithm downwards. Yeah. Yeah. So for my, so so this is like a personal taste. So I've never felt accommodated with this TensorFlow and Keras stuff, because it was, or might be to my limitations, but it's like was like too far away, like to inject this graph structure and this with this. So and So the things we are using are much more like normal algorithms you work with. So not with this graph structure and so. But it's maybe it's a kind of flavor. But I think it's more. So the advantage, in fact, is from our method is that you can really easily and really easily, because I can say that I'm not a good programmer. You can really easily try to adapt or to modify this, and they really, as the title says, accessible. And I think this is
yeah. And actually, to that note, I think there's a very good question by Michael Mannerung. What learning resource would you recommend to a biologist who are interested in deep learning? Many examples out there, but many have emphasis on image recognition. So what about like other data?
What actually is a good resource, I guess, is this deep learning book from Ian Goodfellow, where all aspects are covered. And I didn't know if I get it right. So learning resources from like data or about the.
I think we're learning about deep learning.
Learning about like then this deep learning book of Goodfellow. I can also copy it in the chat.
Yeah, that would be useful.
And so what actually also is very nice, so it's but more like learning by doing, is what I didn't mention is there's this flux like a similar to TensorFlow, but much more easier or much more related to normal, I would say, normal programming stuff. And this is also we highly recommend, because there's a called Model Zoo, where you have different models pre-configured and with image data, and you can try them out. And so this is really nice to play with the models. And I can also copy it.
Perfect. Yeah, that's very useful. So back to Federico. Let's see if we have more questions for you. I'm sure. Cristina Cheroni asks if GeneTonic is also extensible to the exploration of gene set enrichment analysis beyond classical gene ontology enrichment.
Yes, these shaker functions that I've been writing lately are actually what could do the job for you. So there is a shake_FGSEA. So if you're using FGSEA as a framework in Bioconductor, then you might be just say plugging in the output of that pipeline and use that in GeneTonic. There are some requirements on the columns that might be present, that has to be present to actually display the content correctly. So, for example, I'm using the leading edge information as if they were the differential expert genes that would be annotated to that particular gene set. But I think that's a workaround that is reasonable to work with.
All right, and Lucio Palazzo asks if it's possible to use gene expressions with time-dependent features.
I think so. I mean, in the end, it would just so the principle would be that you do all the analysis first. So you have your result for differential expression. You have some kind of list of genes that is, say, tabularly based, coming out of this DE computation. So ideally, yes, you could. So probably it would make sense to structure your data in such a way that whatever heat maps you might get out from would somehow have the order of the columns might be already meaningful. Otherwise, say you can do some pre-processing in advance and then you just plug them in.
All right. Makes sense. I have a question here addressed to Mattia Chiesa in my document, but it might be good for Moritz as well. The question says that deep learning can work with some data set and sometimes with not others. I guess that's true for any classification or any algorithms, actually. But it can also be quite computationally demanding. And I guess this could be a question for everybody, really, like how are your package making deep learning more computationally efficient? So I'm guessing that actually the question is for Moritz. You're muted, Federico.
No, so I have to first. So actually, so I'm only the user here. So the package actually are developed by Stefan and Stefan Lenz, which I also mentioned. Especially the Boltzmann machines package and the Julia Connect that use it in R. So, but as a premium user, maybe I can state that that it makes it more. As the title says, more accessible. You can it allows you to really play and really work with the algorithms. I think better than in the conventional frameworks. Yeah. So for me, that's it.
All right, I think we're out of time. So would like to thank again all of the speakers of this session. I think it was a fantastic session. And also I would like to thank all of the attendees that asked questions. It's good when you're a chair and you don't have to come up with your own questions, because it means that the talks were interesting and people were engaged, and I think it's a win. So I guess I'll see you all at the other sessions, and enjoy the rest of the conference.
Thank
Bye.
you. Goodbye.
Thank you. Bye.
Mm.